Hello, my name is Kevin McLemore, and today we're going to be doing a critical review of the primary source parallel reaction monitoring for high resolution and high mass accuracy quantitative targeted proteomics. So let's look at an overview of the primary source. Parallel reaction monitoring, or PRM, is a new method for targeted proteomics that use a LC or HPLC, however you heard the instrument called. This allowed for increased dynamic range and reduced method development time. This study compared the performance of PRM to selected reaction monitoring SRM on yeast database. PRM was able to detect a wider range of protein than SRM with a dynamic range that was up to 100-fold greater. PRM also required less method development time than SRM. These results demonstrate that PRM is a promising new method for targeted proteomics. PRM offers several advantages over SRM, including increased dynamic range, reduced method development time, and automatic data analysis. Some limitations of PRM is sensitivity and has a lesser of ability to analyze complex samples over SRM. So if there is a already established monitoring system to analyze protein, what would be the need to try to improve on it? Well, selected reaction monitoring SRM is a widely used method for targeted proteomics. However, SRM can be limited by its narrow dynamic range and the need for extensive method development. Here we present a new method for targeted proteomics called parallel reaction monitoring, abbreviated PRM. PRM use a quadpole equipped high resolution and accuracy mass instrument. This is important because it can simultaneously detect targeted product ions in one concentrated high res analysis. This allows for increased dynamic range and a reduced method development time. So the basis of this primary source is PRM can simultaneously detect all targeted product ions in one concentrated high resolution mass analysis. So here we have two spectrums. Here is the new PRM and here is the SRM. Here you can see on the bottom is your retention time and going up is your concentration. With PRM, you can see with the concentration that you have more analytes being analyzed, and for the SRM, over a certain amount of time on your retention time, you see them coming off at different intervals. And down here, again, you can see um, that at a different retention time, you can see that the analytes with the PRM is coming out grouped together, and with the SRM, you see them being bounced around, being analyzed at different times. So this spectrum allows us to see exactly how these analytes can be analyzed at a certain concentration all at once with the new PRM and not scattered along this retention time at different concentration with the current SRM. So let's talk about the major findings from the results of this primary source. The first finding, increased dynamic range. PRM was able to detect a wider range of protein than SRM with a dynamic range that was up to 100-fold greater. This means that PRM can be used to detect proteins that are less abundant, which is important for many proteomics applications. The next finding was reduced method development time. PRM required less method development time than SRM. This is because PRM only required the optimization of a single parameter, while SRM required the optimization of multiple parameters. Finally, automatic data analysis. PRM can be used to automate the data analysis process. This can save time and resources and it can also help to improve the accuracy of the data analyzed. So let's look a little bit at the experimental method. The instrument that's used in this experiment is a LC, or a liquid chromatographer. It's two very important components to this LC that was being used for this PRM experiment. You have the cropole analyzer and the mass spectrometer. The cropole allow for the identification and the quantification of multiple protein in a single experiment, and the mass spectrometer was necessary to fragment the peptides and identify them by their mass and fragmented patterns. So for a moment, let's look at the LC or HPLC, however you heard it called before, at its components and exactly how the analytes actually get analyzed. So in an LC, of course, you have the liquid parts, liquid chromatography. You usually have your mobile phase, you have your column, and you have your spectrometer. So, and you have your detection system. We know that this is a mass spec. This is where it's going to, this is the spectrum where it's going to actually get broke down and fragmented to be read. So what actually happened is your mobile phase carry your analytes through your column. Depending on your analytes and the packet in your column, is going to come off at different times, and these times are called your retention time. So each analyte have a unique fingerprint, and it's going to come off at different retention time, and depending on where it comes off on its retention time, you're going to be able to recognize that component. So we know that this LC is equipped with a quadrupole mass analyzer. So what exactly is happening in this quadrupole analyzer? A quadrupole mass analyzer consists of a set of 4 conducting rods arranged in parallel with a space in the middle. The opposing pair of rods are electrically connected to each other. This is a type of mass analyzer separated ions based on the stability of their flight trajectory through an isolated electronic field in the quadrupole. The field is generated when a radio frequency or F voltage is applied between one pair of the opposing rods in the quadrupole. So once again, this is what happened once the analyte go through those tubes that was just discussed. It's going to go through the column, and as we stated before in the previous slides in this presentation, this column is packed with a certain type of packing that's going to react with those analytes. And again, when it goes through the detection software, it's going to come off on the chromatogram at some retention time, and the retention time is going to be correlated with the resident time and the elution time as those analytes are getting hung up or slowed down going through that packing. Another major spectroscopy that was used in this primary source was the tandem mass spectrometer. The tandem mass spectrometer is based on coupling mass spectrometers together in a series to analyze complex mixtures. The method used 2 mass filters arranged in sequence with a collision cell between them, and the collision cell, the precursor ion, collide with the gas molecules and are fragmented into smaller ions referred to as product ions. So now that we have looked at the instrumentation that was used, and we know the analytes and how they played a part in this experiment, so how do we know that the results that was generated is actually statistically correct? The data that were analyzed by the use of unpaired t-tests with Welch's correction for equal valences to assess the statistical significance of their results. This is a standard statistical test that is used to compare the means of two graphs when the variance of two groups are not equal. The data in this experiment showed that the results had a p-value of less than 0.05. Why is this important? The p-value is a statistical measurement that is used to determine the statistical significance of a result. The p-value less than 0.05 is shown to be statistical significant, meaning that there is less than a 5% chance that the result could be occurred by chance. So this let us know that the results that was coming off of those instruments wasn't happening sporadically or by any kind of chance, that the result was statistically accurate and correct. Versus correction for unequal valences let us know that the statistical method that we use to adjust the p-value of an unpaired t-test when the valences of the two groups are not equal. This is done to account for the fact that the p-value may be too low if the valences are not equal. So let's look at the results. The purpose of this study was to determine the effect of a new treatment on protein expression using a new instrument, the PRM. One group that received the treatment and one group did not receive the treatment. The author found that the mean protein expression level in the group that received the treatment was significantly higher than the mean protein expression level in the group that did not receive the treatment. This suggests that the treatment had a positive effect on protein expression. Now that the results are in, that look at some limitations that could have had some impacts on the results. One limitation was small sampling size. The study only included 30 patients, which is a small number of participants to draw any firm conclusion on. Relative protein expression is another limitation. The study only measured the relative level of protein expression, not the actual level of protein expression. This means that it is possible that the difference in protein expression between the two groups of cells was not clinically significant. Other factors can be patient's age, overall health, and their treatment history wasn't tightly kept, or it wasn't a big enough group to actually um, account for those things because it was a small sampling size. So how can we control or eliminate limitations in the future? We can have a greater sampling size. Future studies should have a larger sampling size. This will increase the statistical power of the finding. We can look at the actual protein level and not just the relative levels of the protein expression. Other factors that we can control is the participant age, health, and treatment history. We could try to group them together or tightly control that factor. So what are some proposed future experiments that we can look at? So we can improve the sensitivity of PRM. We know that one limitation of PRM is that it is not as sensitive as SRM. Another experiment we could look at is develop methods for using PRM with complex sampling. One of the limitation of PRM is that it had a difficulty when using complex samples. Another proposed future experiment is to compare PRM to other targeted parametric methods. PRM is a relative new method, and it has not yet been widely compared to other targeted methods. Applying PRM to different biological samples can be another experiment looked at. Overall, the study provided promising results for the use of PRM in targeted proteomics. PRM offers several advantages over SRM, and it has the potential to be more effective and accurate method for targeting and detecting qualifying proteins. Here are some of the key takeaways from the study. PRM is a new method for targeted proteomics that offer several advantages over SRM. PRM was able to detect a wider range of protein than SRM with a dynamic range that was up to 100-fold greater. PRM required less method development time than SRM. PRM can be used to automate the data analysis process. There are some limitations to PRM that need to be addressed, such as its sensitivity and its use of complex sample. Future work could focus on improving sensitivity of PRM and developing methods that use PRM with other samples. Here are the references that was used in this presentation. Once again, thank you for your time.